continuation of what I delivered last week. That was on genotyping of plant pathogens and agriculturally important microorganisms. This lecture is for half an hour or maximum 45 minutes. We will discuss something on molecular phylogeny that uses allelic variations in the gene sequences rather than the sequence data per se. Let us see how this genotyping tool works, how to use these genotypic tools in our day-to-day population genetic analysis. This morning we heard the practical about molecular phylogeny. That molecular phylogeny exploits sequence data. I must emphasize here, it is sequence data, because you are comparing the gene sequences or sequences of genomic region wherein you expect some variations or no variation, either way. So, you do molecular phylogeny after doing sequence alignment. Okay. Hope that is clear to all of you. If anybody is not clear about it, one or two was missing this morning, you really missed it, whoever missed it. Nevertheless, you can catch up in the coming days. The tool what I am going to teach now, which does not exploit sequence data directly, right? It is going to exploit some numerical values that is assigned to the sequences which is showing some kind of variability. Okay. Allelic variation based molecular phylogenetic analysis, also called as multi-locus sequence typing. Let us see how to do this multi-locus sequence typing in microbiology and plant pathology. Right. I just borrowed these slides from a very nice website, appliedmaths.com. I think please visit this website. I just brought those information. It was very good information for MLST. Multi-locus sequence typing is a molecular typing technique whereby a number of well-chosen housekeeping genes are sequenced, usually in part. Meaning, we do not sequence the entire gene, rather we are sequencing only partial gene. Suppose the gene length is around 2 kb. We do not sequence the 2 kb, rather we sequence only a part of it, number one. With respect to plant pathology, we also consider some virulence genes. So, it is a combination of housekeeping genes and some virulence related genes or virulence factor coding genes, okay, for this multi-locus sequence typing. In a typical multi-locus sequence typing approach, what is exploited is the recombination is expected to occur with a much higher frequency than point mutation. This is precisely exploited in multi-locus sequence typing, right. Therefore, one does not look at the total sequence similarity between strains. Instead, each sequence of a given locus is screened for identity with already known sequences for that locus. It only implies that you need to refer a database where the sequences are already uploaded, right. If the sequence is different, it is considered to be a new allele. The question here is, what is the difference? What do you mean by difference here? So, it can be a small recombination event, or it can be a point mutation, but we expect more frequently the recombination than point mutations here. And the new allele is assigned a allele number. I will tell you precisely how to assign this allele number. Usually, we consider 7 loci from the genome. The choice of these 7 loci is so critical for the success of multi-locus sequence typing. You have to select those genes which is having housekeeping function, and that has got a species level signature. That is very, very important. So, in case 7 housekeeping genes are studied, each strain is thus characterized by a profile of 7 allele numbers. You got it? You are going to consider 7 genes or 7 loci, right? I must tell you here, this 7 loci is sequenced, right? Sequencing is the fundamental. You have to sequence those 7 loci. After sequencing the 7 loci, you are going to compare it with a well-established database where these loci sequences are already available for you. I will tell you how many different databases are available which is housing the loci which is meant for multi-locus sequence typing. The allelic profile, what do you mean by allelic profile? The string of numbers which is assigned to each and every loci, right? Gene 1, gene 2, gene 3 up to gene 7, you may call it as locus 1, locus 2, locus 3 up to locus 7, and each locus is sequenced, right? You understand? And remember, the length of the sequence for each and every loci may be different. It need not be same. Maybe loci 1 or gene 1, you have a length of around 600 base pair. Loci 2 may be 1200 base pair. Loci 3 may be even as low as maybe 300 base pair. So, that is your choice. That is decided by the conservation or the conserved nature of that particular loci. Okay? So, each and every loci is sequenced, assigned a specific allele number, and you stitch all those allele numbers together. It becomes, if it is a single digit allele number for each one of those loci, it becomes a 7-digit number. That is called allelic profile. Okay? In genomics terminology, we call it as sequence type. It is very popular in medical field, STs. ST stands for sequence type. That is nothing but string of allele numbers assigned to each and every sequenced loci. Okay, the critical question here is how to assign the number, right? What are all the criteria to assign the number? Number 1, for that you need a reference database. Okay? And MLST has been used successfully to study population genetics. If somebody is studying population genetics of microorganisms, population genetics of plant pathogens, right? The population of a given pathogen. That means what? You know the identity of that organism well in advance. You know what is the species in question. Right now, you want to go for population genetic analysis. So, here typically used in population genetic analysis. Okay? You can reconstruct microevolution of epidemic bacteria and other microorganisms. So, multi-locus sequence typing has got relevance for bacteriology in general, and more so for rapidly evolving or well established bacterial pathogens of plants or even human, right? In fact, plant scientists, we have adapted this tool from a medical field. Originally, this multi-locus sequence typing was developed for some of the dangerous human pathogenic bacteria. Okay? I will take you how it works. This is a snapshot of how this sequence variation is converted into a simple integers, numbers. Okay. You can see here, this is the loci. For instance, strain A has got, which I already mentioned, there are 7 loci. Somebody has selected the 7 loci starting from A, B, C. I don't want to read it. You just pay attention to the first one. They have assigned the number 1. Okay? And just I'll draw your attention to the strain B, or the same loci. They have assigned the number 2. What prompted the investigator to assign the number 2 here? The difference of a single nucleotide variation. In a stretch of around 600 base pair sequence, they found somewhere in the middle or somewhere in the edge or side a single base pair variation. Instead of G, they found C. So they made a multiple alignment again. It is the same multiple alignment, right? So they found, instead of G, they found C. Rest all same. So they, instead of assigning the number 1, they assign the number 2. So this is allele number. You come to strain C, they found again G. They assign number 1 again. Okay. Move on to the second loci. They found a small stretch of 4 nucleotide base pair variation at a stretch. You pay attention to that. They consider the entire 4 nucleotide bases together, and the entire length they found. Wherever they found A, C, T, G, they started assigning the same number. If there is a small difference, they started giving different number. So basically, the number is assigned after doing multiple alignment. Okay, the number of base pair variation is assigned as unique number, that is called as allele number. Now move on to the strain B. For the same gene, they found CCAG. They started giving the number 2, and the strain C, it takes the number 3. So likewise, either the nucleotide variation at a single base pair level, or 2 nucleotide level, or 3 nucleotide level, or even 4 nucleotide level, it is assessed and the numbers are assigned. This is clear to all of you? Now, if you stitch all the 7 genes together, the first strain is characterized as 1, 1, 1, 1, 1. If you move on to the second strain, it is 2, 2, 2, again 1, 2, 1, 2. Right? If you move on to the third, again 1, 3, 2, 2, 1, 3, 3. What does it reveal actually? It only reveals how many of the alleles are shared among the members of a particular population. You understand? For example, some alleles are shared by all, some alleles are shared only by 2 of them, some alleles are not at all shared. So this is a very complex, matrix you can create. Suppose if you imagine you handle 7 or 10 loci, 100 entries in a population, and it's really a matrix of numbers. Right? I'll ask one question. How many number you can accommodate? Anybody? Loci length is around 400 base pair. How many possible alleles? Pardon? The figure is really mind-boggling, right? Each base can be potentially any one of the 4 bases, correct? It's really a huge number comes in end of the day, right?
On the strain C, the last one, second last, means all strain A, strain B, we have given 1, 1.
Yeah.
And why in the third it is 3? It should be given as second because the
No, because they considered 3 bases, you know. TGG, again TGG, they take same number. And it is GAA. Nucleated variation at 3 bases they considered.
no, that I am getting, but
So-
other cases also, no? Like
That's what I am telling. Nucleated variation at single base pair level, 2 level, 3 levels, and 4 levels. That means all possible nucleated variation is accommodated here. Right? I'll go with a typical example. You will get much more information on that. So, a plant pathogen like Magnaporthy or Ralstonia, right, or Xanthomonas, you can do the same work. So, what you have to do? You get into the genome data and shortlist the potential housekeeping loci or housekeeping genes. Sequence it. If you are doing it for the first time, you are going to assign the number for the first time. Right? If reference sequence is already there, if the reference sequence was not assigned allele number, then again you are going to assign the allele number for the first time. So, let us see how we went ahead of this particular work. We generated the whole genome sequence data. All right. Now, when we analyze the database, we could find around 189 isolates, which is already whole genome sequenced. You understand? So, you have a population already. So, 189 whole genome of Magnaporthy is already there. So, now we have to select the loci, and here we have considered around 17 loci. 10 of them are housekeeping and 7 of them are virulence related, or what is called as pathogenicity related, or effector coding genes. Now, what you do? Variation at nucleated for each of the loci was assigned unique allele number, the way I explained in the previous slide. So, prior to that, you generate the sequence information of the test isolates what you handle in your laboratory. So, we handled around 24 isolates, right. They were collected from a specific single field representing different tissues, right. The genomic DNA was isolated, primers were designed from the whole genome, right. Primers were validated and sequencing was done. So, once sequencing was done, now you have the data set in your hand. You have 24 data set in your kitty, and you have 189 data sets from public domain, right. In total around 212, in total a huge population of data sets. Now, we made an attempt. Can we assign allele number? So, what is needed? The first criteria is you have to normalize the sequence. What do you mean by normalization? You have to do end trimming, good base calls. Good base calls are assessed based on the peak area, peak size, quality values, everything was done, right. So, you have the sequence in your hand now, curated sequence. For every specific loci, the sequence length is trimmed and kept constant. This morning we talked about it. The first base is representing a specific coordinate that is constant for all entries. Not only the 24 entries what we handle, but also 189 entries what was available in the database. Okay? All curated sequences are now normalized. Maybe the length varies across different loci. Okay? Now, the string of allele numbers, we call it as sequence type, also called as allelic profile. And the allele data was subjected to a completely different algorithm. It is not the one we discussed this morning, that is MEGA. This is a completely different algorithm, that is what is called as E-bust algorithm. All right. Now, the population genetic tool you can come out with different kind of data interpretations. Let's see what kind of interpretations we could make it here. Up to this point, is it clear to all of you?
Sir, allelic number whether manually you are doing or with the software?
It's given manually. There is no software involved here. That's the beauty of it. I'll take you how to do it. Somebody was asking how to give the allele number. So here comes the slide. It's only a kind of a hypothetical example. For example, you have x1 to x8 are different isolates. Okay, let's say it's a magnoporthy here, or it can be anything. And you have a particular gene, right? Remember, a particular gene, right? Let us say this gene is around 600 base pair length. I am only showing the first 6 bases, how it behaves. Remember, this is only a hypothetical example. Take the example of isolate 1. The first base call, it is A. Isolate 2, the base call is A again. But if you see the isolate 3, you are seeing the base call T there, right? So we gave 1 here. When I move to the isolate 2, I found the second base call. Had it been T, I would have given 1 here. But I found A here, right? Remember, it's the first 6 base pair. Variations may come later on also, right? So now, these variations are converted into allele numbers. So I gave 2 here. When I move to the third one, I found there is a variation in the first base call, right? I gave 3. Why 3 here? It is different from first and second. That's it. Okay. When I move to isolate 4, I could get exactly the same like the first one. Then that prompted me to give again the number 1 here, not 4. Okay. Then I moved to the 4, I started seeing variation in the 5th base call. Then I gave number 4 here, right? And again, when I moved on to the 6th isolate, I found there is variation in another region. So I gave number 5 here. Then 7th one, again, it's completely different from the previous 3. And again, it is different from the previous 3 also. Then I started giving
X5, X7 and X8 are
okay, fine. These two, again you take the 4. So what I mean to say is the nucleated variation, what is coming within the gene sequence is assigned allele numbers. Okay. This is another, you move on to the gene number 2. Do the same thing. So remember, this is done by multiple alignment. Nothing is done manually. If you do manually, you may commit mistake. You cannot score it. It's very easy, very difficult. With this 6 base, we are getting confusion. If it is 600 base, you can imagine how much confusion is there. Now, end of the day, you have to assign alleles for all loci. So come back to our own project. When we started doing this assigning the allele number, this particular genome was originally available in the database and supposed to be the first genome. In 2004, it was sequenced. So what we consider this as the first genome of magnoporthy. So there is no other option. You have to give 1, 1, 1, 1, 1. Why? Anybody? There was no reference. This is going to be the reference. This is very important for all of you. He was asking who will assign the number. It is we who is going to assign the number. Suppose such a data is not available, whatever may be the organism, in particular bacteria you are handling, right? Now what you have to do? If it is for the first time, it takes the number 1, right? So we assigned number 1, 1, 1. This becomes a reference now. And this is an isolate number, and these are all the genes, the 17 genes. That is 10 housekeeping genes and 7 virulence genes. You just give 1 to all if it is described for the first time. Now let us take you to how to analyze this data. Now you have a mix of numbers now. Okay? Maybe I can come back to this slide once again. Before that, I just tell you this is the data what we assigned. So 70-15, it takes 1, 1, 1, 1, 1. These are all the isolates, our own isolates, retrieved from databases, and each one of the gene is multiply aligned. After seeing the variations, we started assigning the numbers, right? So this is really very confusing numbers, all are numbers only. Remember, 212 entries, 17 loci, it's full of numbers. Now how to analyze this data? Can we convert this data into a kind of matrix? Possible? It's a matrix only, right? So what we do, we put all the numbers together in Excel sheet, right? The size of the Excel sheet is row versus column. Okay, number of entries is equal to R into C. Suppose if you have 200 rows and 17 columns, you just multiply it. So that many numbers will be there in that Excel sheet. Okay. This is just to let you know that how to assign the allele numbers. Remember, allele numbers are assigned after verifying the trace file. This is one of the most important criteria. It is not the sec file what is provided to you by the private company is considered here. What is considered here? Only the trace file, right? The trace files are checked for base pair variations. We discussed here how to use Finch TV and the CLC together to call a base. You understand? So that is the guiding force here to derive at this matrix. Okay? And that's what it is given here. You have a genomic DNA, you have PCR products, allele sequences in trace file, and allelic profile. Okay? Then this is what is called as sequence type. I'll take you. This is a matrix. It goes like this. This is the number of entries. Again, it goes like this. This is input data which you can take on board for MLST analysis. I told 212 isolates, right? But if you see the previous slide, we reduced to a certain number, and that is highlighted here, 48 distinct sequence types. What is the meaning of it? Distinct sequence types. That means number of isolates, they shared all the alleles. You got it? The isolates which shares all the alleles are considered to be clonal. They are clones. So the point is, the duplicate entries takes only one sequence type. You got it? I just welcome Dushyant. Please take your seat. Let me complete my class. Yeah. Now, how to do that? In a maze of numbers, how to remove the duplicates? This is something like this. You do not know in a snapshot. You are not able to see how many of them are similar, how many of them are different. Softwares will help you. Okay, so what you have to do is all 212 data into the loci, 17 loci, the huge matrix, you just input. It's an input in your software. So how to do that? Create a matrix. This is a matrix. right? This matrix is just imported in Excel sheet. In Excel sheet, in your first column, it is sequence types. Remember, first column is sequence type, and rest all alleles. You will find around 17 alleles here. Okay, I mean 17 loci. I'm sorry, 17 loci. Now, once you cut paste into Excel sheet. This morning we were talking about different formats. It's not that every format is going to be accepted by the software. The format which is accepted in MLST data analysis software is Excel tab delimited format. Okay, you will be practicing this in the coming days. Cut paste in Excel sheet, save as tab delimited format. Once you do that, it will be something like this. You understand? And this becomes an input data in your MLST analysis. How to do that? You will find the software in this particular link. You just Google it out, eBust v3. Okay? We will demonstrate. So you just click it, Windows will open. There will be a place where you can upload your data. What data you are going to upload here? Excel tab delimited format data. Okay, just give insight and run the program. It is so user friendly. You have to learn by doing. Okay. Once you do that, it will give you an image something like this. Okay. And this image reveals how your population behaves. I already told two isolates that is sharing all alleles are duplicates. They are clones. Two isolates, they are sharing all alleles except one. Maybe they are twins but not identical twins. Correct? In an analogy. Right? Two isolates, they share all except 3 or 4. They are closely related, maybe they are relatives. Is that clear? If two isolates, they share only 50% of the alleles, the rest 50% of the alleles are completely different. That means these two individuals shared a common ancestor, right? They evolved maybe in the last 100 years. Two isolates, they are not at all sharing any alleles. That means they are completely different entities in the population. They traces a different parentage or different lineage. Right? And I must tell you that you will count around 48 entries here. Right? I'm sure you cannot create a phylogenetic tree based on sequences by accommodating 48 entries. Right? Maybe the coming days I will show you a eBus tree which is having 1003 isolates. Still you are able to make some interpretation about the data. Right. The point here is this tree must be visualized in a different manner, and there is something in the middle and the radiation is happening. Right. The one in the middle is sharing maximum number of alleles with the rest all. The one which is sharing maximum number of allele, what we consider is the founder of that particular population. You got it? You are tracing the origin of the isolate. Right? The isolate which gave birth to other isolates, and they had an independent evolution later on. Right? They evolve. So, in a population genetic analysis, this allele data by eBust algorithm is one of the most valuable tool, right? So, I am not getting into the finer details of this this particular tree. The purpose of this lecture is only let you know how to handle the allele data and how to convert the allele data into a phylogenetic tree which reveals the behavior of the population or population genetic analysis. Okay? So, of course, these are all some of the interpretations we made, much similar to the interpretation what we got in the previous analysis. Paddy-blast isolates are clustered together, and we have finger millet and pale millet. They were singleton in the population. Of course, this is some of the interpretations. This is in continuation of my last week class. We are trying to develop a new genotyping tool based on whole genome sequence data for population genetic analysis of this blast fungus, and all 212 isolates clustered into 48 distinct sequence type for allelic profiles, which indicates the clonal nature of the pathogen. Again, if all of them are sharing all the alleles, that means population is not evolving, and it is what do you call, there is no variability at all. But rarely you will encounter such a scenario. There is always some variation will be there. And genotyping tool clearly revealed rice isolates in one group with the finger millet and pale millet isolates were emerged as singletons, and these isolates shared very few alleles with rice infecting blast fungus. And of course, all neck and leaf blast isolates, they shared all the alleles. Right? So, you can make some robust interpretations and strong conclusions. Right. With that, I will take you to a tour of very useful databases which you might be interested. Okay? Up to this point of time, if you have any quick questions, you can clarify. We can have in the late evening also. There is no problems on that. The next speaker has already arrived. Yeah. Any? Can you take the mic and ask? In a while, there is some similarities, right? We have not done the principal component analysis for this data set, so definitely I cannot answer your question directly now. Yeah. I'll take you to the databases of some of the very popular MLST data. This is what is called as one of the very old database for MLST, right? You can just browse it. That will take you to different MLST databases. They houses a different set of organisms, right? UK one, there is a MLST database from UK. I think there's a pub MLST. That houses most of the human pathogenic bacteria, in particular Pseudomonas aeruginosa. Thousands of entries are there, right? And here also you will find more of human pathogenic bacteria, right? You can see here Pseudomonas putida, different kind of human pathogenic bacteria. This is the one which we are interested. This database is for plant associated microorganisms database, also called as PAMDB. This houses some of the plant pathogenic bacteria. You will find Xanthomonas, you will find Pseudomonas, you will find Ralstonia, Azidovorex. Okay. So the question is, what these databases really do? What is their function? How does it work? I must tell you that These databases are maintained by individuals. Right? PAMDB is maintained by Boris Venatzer in Virginia Tech. Okay? Much similar to that, you have PubMLST is maintained by scientists in UK. So what they do? They collect data sets from all over the world. For example, PAMDB, you can submit your data. Right? And what kind of data? Here comes the twist here. Unlike GenBank, these databases insist you to submit trace files. They will not accept sec file. You cannot submit sec file. You have to submit trace files, because trace files are original data files. Right? You have to submit trace file with all possible data with annotations. All right. And the database curator, what he does? He sees the trace file, he analyzes it, and he retrieves the sequence data from the trace file. And you can compare that data with the one which is given to you by the service provider, the genome sequencing service provider. And we found number of times there are differences, at least a single base pair variation differences. Once it is analyzed by the curator, now your data is accepted for uploading in their website. Right? Then it is available for anybody. And second thing what they do, they analyze the trace file, retrieve the data. After retrieval, the data is resized for the purpose of documenting in allele database. Right? Did you understand this point? Not exactly. For example, if loci size is around 800 base pair, but the consensus sequence what is needed for assigning the allele number is, let us say, 650. Right? The only 650 nucleotide base calls are documented. Right? And this 650 is constant for that particular loci across all the entries. You got it? I do the sequencing, I got 800. You do the sequencing, you got around 700. Somebody else does 750. Yet another people who is doing 550. Minimum length what is needed here is 650. The one who did 550, he will be asked to repeat the sequencing, because the length is short. That is not accepted. So that is the beauty of this database. That means all sequences what is uploaded there, they are same length, same base call. Right? So that goes back to my original slide, what I told. MLST database, the allele numbers are assigned based on pre-existing curated data. Right? Tomorrow if you want to claim that we have done a database for Magnapurthi, what I need to do? I need to create a database and upload the data sets and put all the entries there. Tomorrow somebody else will do, they will match our data, then start assigning the allele number. Right? Remember, assigning the allele number is your job. The critical question here is, if it is already existing database, how to assign allele number? It's very easy. For example, if somebody wants to do Ralstonian, you don't have to bother too much. You just sequence it, and you know the minimum length of each and every loci. If you have that particular length in your hand, you just blast in this database. If the length is enough, it will immediately tell you the isolate what you are handling, a gene 1 of that isolate is sharing all the sequences with a pre-existing data. So it will pick up the allele number from the previous data. You got it? and it will tell you your gene sequence is having 100% match with the gene 1 of so and so isolate. Allele numbers are instantly assigned. By chance, if there is a variation of single base pair, then only the question comes. So, who will assign the allele number? It is not you, it is a curator. He will ask, okay, you blasted it, you found variations, then he will send a mail to you. You send the trace file to me. Then he will analyze the trace file. Then he will say, okay, you made a mistake. Actually, there is no single base pair variation. It is same as something already there in the database. He will assign that particular number to you. By chance, if that allele number is really the base pair base pair variation is really genuine, then he will check the trace file and he will say, okay, you found a new allele for that particular loci. You got it? The new allele number is the next number of the number what is already documented there. For example, if 49 alleles are there for a loci ADK, it's already there. So, your sequence is not matching with any one of those 49, correct? Then he will give allele number 50. So, this is what is called as participation of everybody in building a database. You understand? So, the population, the database expands, right? So, maybe today it is 49 allele, maybe after 5 years it is 120 alleles. It goes like that, right? So, people felt when the database expands, we need better and better visualization technology. So, eBust has come very handy. It can accommodate thousands of entries without any problems. You got it? So, with that, I will conclude this genotyping of plant pathogens and agriculturally important microbe with a special reference to multi-locus sequence typing. Okay? Remember, multi-locus sequence typing, experimental pipeline is slightly different from the conventional molecular phylogeny pipeline. We take a deviation from the multiple alignment data, right? We will not use directly there. Instead, we convert that variation into allele numbers. Okay. So, I think with that will let me conclude. Any doubts before we? Yeah, please take the mic, please.
How do we know MLS in case of multiple alleles?
Multiple?
Alleles, if multiple alleles are present.
Anybody can answer this question? We are characterizing the multiple alleles only here.
Sir, but we are having a different, we are seeing that how it is different with different organisms. So, but if the same organism has multiple alleles, then how it is?
Multi-copy you are meaning?
Yes.
You mean multi-copy? You take both of them on board, no problems. Yes? Anybody? Any more questions? Hope I made it very simple, right? But you have to practice on your own. Okay? Yeah, Kuleshwar.
Two things I want to ask. One is suppose the reference genome what we are considering that we are giving all number 1 allele, and suppose I have given a set of allele with a certain isolates. Then 5 years later somebody else is studying the same thing, and if he is finding maybe some new alleles or the existing isolate as a duplicate copy, then he will assign the same allele number what I have given, or he will assign a new number.
No, that's what I already clarified this point. What you are asking is after 5 years, you are isolating the same organism means what? because the 5 years this organism was exposed to nature. If some variation happens because of that, if allele number happen to be different, then it takes a new number. That is all about how the pathogen or how the microorganisms evolve with time. You understand? You got my point? So, who will assign the allele number if the database is already there? If it happen to be a new allele, the new allele is always assigned by the curator, right? The old allele numbers are assigned by you after aligning with the existing database. Okay, hope I answered your question. Yeah. Okay, I think I'm happy to see that a lot of interest is coming. So what we will do, we will keep on discussing this in the coming days. Now I will stop my, I may call it as a class or lecture, whatever it is. And so let me stop here, and we will move on to the second agenda this afternoon. Okay. Madam is coming. Okay, okay. Yeah, fine. Okay.